So I'm going to talk about some simulation work, some polymer simulation work, which I've sort of been developing over quite a few years now, looking at chromosome organization, and particularly more recently, I've been looking at chromosome organization in cancer. And so obviously there's a lot of biology involved here, and I don't have time to talk about that in too much detail. So I thought I would instead try and just give you a sort of an overview to give you a flavor of what it is that we're trying to do. So rather than talking in detail about the results. So I'll start with a brief introduction on the biology for people who aren't so familiar with it. And there I want to convince you of two things. So first, I want to convince you that the three-dimensional organization of the chromosomes within the cell nucleus plays a role in some biological function, and particular genome gene regulation. And then I want to convince you that there's some physics, some physical mechanisms which are going on, which are involved in that. And then I'll tell you a bit about the model we've developed and some of the underlying physics and some of the things we've been doing with it. Okay. So we're talking about chromosomes in humans, and so chromosomes are made up from chromatin, which is a fibrous material made from DNA and proteins. And this is a sort of a cartoon which shows the structure of chromatin. So this green tube here represents DNA, and it's wrapped around these small discs, which represent bundles or complexes of proteins called histones, and these form these disc-like structures which are called nucleosomes. And then this string of nucleosomes fold up into a thicker fiber. And we know quite a lot about the structure of the nucleosomes. We know its crystal structure, but we know much less about how these are folded into thicker fibers. So we take a sort of standard polymer physics kind of approach, and instead of worrying about the molecular details of how these nucleosomes are arranged into a fiber, we just represent that as a simple polymer, so a chain of beads. So we have a bead and spring polymer, and we're doing coarse-grained molecular dynamics simulations with an implicit solvent and very simple phenomenological interaction potentials. So this chain of beads represents a section of a chromosome. Okay, so I said that that chromosome organization has a functional role, and in particular in gene regulation. And one way in which that is true is that gene activation can often be driven by chromatin looping, so loops forming in the chromosomes. So how does this work? Well, if we imagine a section of a chromosome and say we have a gene here shown as this sort of green bar, then for many genes, in order to activate transcription, you need to have some proteins bind and form a complex at the start of the gene, at the promoter. So this would be a polymerase and some transcription factors and other activating factors. And for many genes, that's enough to activate and drive transcription. For many other genes, you also have to have some proteins binding at another site called an enhancer, and enhancers can be far away from promoters along the chromosome contour. So typically they can be up to megabase pairs away. So how does some proteins binding at an enhancer site down here lead to activation of transcription of a gene, a promoter up here? Well, these two things have to come into physical contact and form a loop in the chromatin. And so they form a complex, and then this leads to activation of transcription. So this is a sort of a simplified picture, but this is the general sort of idea which people currently have. So as well as loops which maybe directly drive activation of transcription, we think that there are also other kinds of loops which you could call architectural or structural loops. And so these might be loops between proteins bound to the chromatin, which instead of directly driving transcription of a gene, maybe they just bias particular interactions. So a loop which forms down here might make it more likely for this enhancer to interact with this promoter, and less likely for this enhancer to interact with other nearby promoters. So hopefully you can see from this that the looping structure of chromatin in 3D is a regulator of gene expression, and maybe you can start to see where polymer physics might be able to help us understand this. So we could think about asking questions like, what is the probability that certain loops will form within our chromosome? Okay, so how do we know about chromosome structure in our cell nuclei? So there are several different kinds of experiments which people can do. So you can do microscopy experiments where you put fluorescent probes at specific points on the chromosome and measure their separation, say. But a lot of what we know about chromosome structure comes from a kind of experiment called chromosome conformation capture, or 3C experiment, or the genome-wide high-throughput version of this is called Hi-C. And in these experiments, they give you information about which regions of the chromosome are found spatially close together within the cell nucleus. So usually it gives you an average picture of a population of typically millions of cells. So I'm not going to give you any details about how those experiments work, but I'm happy to talk afterwards if you're interested. But the information you get out of these experiments are typically, they look like maps like this. So you can think of this as a contact map. So I'm plotting the position along the chromosome on both axes, and then the color at each point tells you how often 2 points on the chromosome were found spatially close together or interacting within this population of cells. So the color at this point tells you how often this point on the chromosome was interacting with this point on the chromosome. And you can see there's a dark stripe down the diagonal, and it gets lighter as you move away from that. And that just reflects the polymer nature of chromosomes. So if you imagine 2 points which are close together along the contour of the chromosome, they're more likely to be close together in 3D space than 2 points which are far apart along the contour of the chromosome. So these are quite complicated experiments, and so there are a number of limitations and drawbacks. But just to point out 2 of them, one is that they're very expensive. So this involves lots of DNA sequencing, and that costs a lot of money. And another limitation is that you need a large sample. So as I said, typically we're looking at a population of millions of cells. And so if you're thinking about human tissues and in particular tumor samples, it's not always possible to get enough cells to do these kinds of experiments. So one of the aims of our modeling work was to say, well, can we use other data to try and predict these maps so that we can do fewer of these experiments. Okay, so another feature, well, another thing which about these maps is that often instead of plotting the whole square, people just cut out sort of the relevant region around the diagonal and rotate it and show it like this. And in this example, you can clearly see that there are sort of dark triangles, or these would be squares along the diagonal. So what are these? So these are known as domains, and basically they're regions of the genome which show an enriched self-interaction. So 2 points on the chromosome which are within the same domain are more likely to be to interact or be together in 3D space than 2 points which are in different or neighboring domains. And the sort of current picture is that this is a way in which the cell can constrain interactions between enhancers and promoters by enclosing them in these domains. And there've been a number of polymer physics-based models which have been used to try and better understand how these domains form, and I'll come back to that in a couple of minutes. Okay, so let's move on to our model. So in this chromatin biology field, if you want people to take notice of your model and remember your model, you need to come up with a catchy name for it. So we called our model the hip hop model for chromosome organization. So somehow this stands for the highly predictive heteromorphic polymer model. And it's it started as a fairly standard polymer physics type model where we have a chain of beads representing a section of a chromosome. And then we combined into it several different physical mechanisms which drive this polymer into specific 3D structures. Okay, so there are 3 sort of main mechanisms, and I'll briefly tell you about each of them. So the first is that as well as the polymer, we also include these diffusing spheres, which represent complexes of chromatin binding proteins, and then we put specific binding sites along our chromosome. And importantly, these can bind to multiple sites at the same time, and so they can form bridges and loops, for example, between promoters and enhancers, and they can also compact regions of the chromosome. And we've been studying this model for quite a few years now. So we also included a second mechanism called active loop extrusion. So we now think that there are proteins within the nucleus which can bind to chromatin and can actively push it into loops using a sort of a motor effect. And so this is a model which has been studied by several different groups in the last few years, and so we incorporated it into our model as well. So this is clearly a non-equilibrium process. And then the third ingredient to the model is we have a heteromorphic polymer. And all I mean by that is that there are some properties of this polymer which vary along its length. And we put that in quite a simple way. So we just have two kinds of polymer. We have some thicker, more compact regions and some thinner, less compact regions. And to do that in the simulations, we just added some additional springs between next nearest neighbor beads in some regions, and that acts to sort of crumple the chromatin, the polymer. And you can think of that as modeling a different structure for these nucleosomal fibers. But we didn't look at that in any detail. Okay, so those are the ingredients for the model, and then we need some experimental data as an input for the model. So the input data is 1D data. So by that I mean it's things like the positions of protein binding sites along the chromosome, or chromatin state data, which tells us which region should have the more crumpled structure and which region should be more open. And these can be obtained from experiments which are typically simpler and easier to do than the high C experiments, which I just mentioned. So then the output of the model is that we generate a 3D structure. So these are stochastic simulations. So we do lots of repeat simulations and generate a whole population or ensemble of structures. And then we can extract things like a prediction of these high C maps and predictions of microscopy measurements. Okay, so is the model any good? Does it work? So here's just a sort of a visual example. So I'm showing some of this high C data from an experiment along the top here, and then underneath, sort of like a mirror image, I'm showing some simulation data results from the same chromosome region, and hopefully you can see that the sort of the prediction for where these domains are is pretty good, and it's not perfect, but we get a pretty good prediction of where the domains are. And we've done a bit more work quantitatively to try and understand how good the prediction is. But given how simple the model is, we think it's a pretty good prediction, which can give us some insight into what's driving these 3D structures. Yeah, so what have we used the model for so far? So we've studied a number of specific genes. One example is a gene called PAX6, which is a gene that's been implicated in a number of birth defects. And so we've used the model to look at looping around the PAX6 gene and how that changes through different stages of development. We've currently got a project where we're trying to simulate all of the genes in the genome in a particular cell line to try and look for similarities and differences between different genes. And then most recently, I've been using the model to look at 3D structure after genome rearrangements, which occur in cancers. And I'll say a little bit more about that in a minute or so. Okay, so these were the sort of three ingredients of the model, and there's sort of interesting physics involved in all of these. I don't have time to talk about all of that, so I'll just say a few things about this first one. So as I said, this diffusing bridge protein model is something we've been studying for quite a while now. And it sort of started with this simple simulation where we just had a sort of a uniform polymer shown in green here representing a piece of chromatin. And we put that in a simulation with these spheres representing some proteins. And we said there's an attractive interaction between the proteins and the polymer, so they combine to the polymer. But we didn't include any attractive interaction between the proteins and the proteins. So quite surprisingly, what you see very quickly after the start of this simulation is that these red proteins are binding to the polymer, and they're also coming together into clusters. And we were surprised because we didn't put any attractive interaction between the proteins. So why do they form these clusters? Well, the reason is because of their ability to form bridges between different chromatin regions. So if you imagine a protein diffuses along and it binds to a piece of chromatin, and then it forms a molecular bridge bringing together two pieces of chromatin, then immediately in that local region, you've got an increase in local chromatin density. So another protein diffusing by is more likely to bind in that same place, and then you have an increase in local protein density. So another piece of chromatin is more likely to bind there. So there's a sort of a positive feedback effect. And you can sort of see how these protein clusters might drive the polymer into specific structures. Okay. And because this is down to the fact that these proteins can form bridges, we call this the bridging-induced attraction, and that's now being shown in vitro as well. So if I were to play this movie for a bit longer and run the simulation for longer, you'd see that these clusters tend to come together and coalesce until eventually you end up with one large protein cluster. But if you look at some images of fluorescently labeled chromatin binding proteins, you tend to see lots of small spots. So these are small clusters and they don't coalesce into one large cluster. So to achieve that in our simulations, we added sort of another feature to the model, which was instead of allowing the proteins to bind anywhere on the chromosome, which was the case in that movie I just showed, we instead said that there are some specific binding sites. So if there are specific binding sites from these proteins, then you don't see coarsening of the clusters. And this is because bridges between these binding sites tend to lead to loops in the chromatin forming, and there's some entropic cost to forming these loops, and so you can't coarsen these clusters into one large cluster. And we found that if you use some experimental data to say where the binding sites for these proteins are, then you can get the system to drive the polymer into specific 3D structures. And actually, you only need 3 species of protein and some data on their binding sites in order to already get pretty good predictions of some of these high C maps. So here we could already predict more than 80% of the positions of these domains. So this is a sort of a mechanism which can drive the chromatin into specific structures. Another feature of these clusters in vivo is that they're quite dynamic. So the proteins within them very quickly exchange with proteins in a sort of soluble pool. They come in and out of the clusters. But in our simulations, once they've formed, our clusters are pretty stable and the proteins stay there for a long time. So we added another ingredient to the model, which was to have the proteins switch stochastically back and forward between a binding state and a non-binding state. And by putting this into the model, we were able to get much more dynamic clusters. So the proteins can now come in and out of the clusters dynamically, but the position and size of the clusters is quite long-lived. So these look much more like the clusters you see in vivo. And I don't have time to go into the details, but this switching actually drives the system away from equilibrium, and it also provides a mechanism for cells to dynamically control these clusters. Okay, so this is I'll just finish now by saying just a couple of words about this work looking at cancer. In many cancers, you often see events where a break in the chromosome has occurred and it's been incorrectly repaired. So, for example, 2 chromosomes might become fused together in a chromosome translocation, and genes near these breakpoints can become misregulated. And within our genomes, we have fragile sites which are prone to these breakage and repair events. And so the same breakage and repair is seen in different cancers. And genes called proto-oncogenes, which are near to these breakpoints, if they become misregulated, then they can drive cancer formation. So with some collaborators in Newcastle, I've been working using these simulations to study specific proto-oncogenes. So there's one example called CCND1, and we're doing simulations looking at what changes after these genome rearrangements, and looking at what different regions are interacting with this gene and what's driving their misregulation. And so now my collaborators in the lab are doing some targeting experiments to try and interfere with the interactions that these genes are having, with a sort of long-term view of using that as some kind of future therapy. Okay, so as I said, this is work which has sort of evolved over quite a number of years. So there's been lots of people involved over the years. So I'll just thank everyone who's been involved, and thank you for listening.
Thank you, Chris, for this nice talk. Are there any questions? Maybe I start. So how is the 1D information you put in related to the sizes or the places of the domains you see?
Yeah, so I mean, there's several different types of data which we use, and I guess the one that mainly drives the 3D structures are just binding sites for these proteins, and these are sort of small, quite small sites of a few, maybe hundreds of base pairs, whereas the domains typically are from hundreds of kilobase pairs up to megabase pairs. So yeah, those sort of small binding sites can drive quite large changes in the 3D structure.
Okay, thank you. There's another question from first Emanuele and then Demian.
Thank you for your nice talk, Chris. I just wanted to ask you a question on the binding proteins. The question would be: do you know or is it known the structure for these proteins, and in particular, if the binding sites, let's say, there are some binding sites on the surface of the protein, or the proteins are overall charged? So, let's say a model which you used is really faithful. I don't know if it made much sense.
Yeah, I think I know what you mean. So most of these proteins have like a specific DNA binding domain, or a binding domain that recognizes the nucleosome proteins or something. So usually, yeah, it would be a discrete site on the protein which binds DNA. But very often they're in large complexes, so they have multiple binding domains, and so in the simulations I showed you, they were always spheres, so they have like an isotropic interaction, and so maybe that's not that realistic, but we've done other simulations where we have more like a patchy protein kind of model where we have discrete binding sites, and so basically so long as you have two or more binding sites, binding regions on the protein, so you need to be able to form a bridge. So if you have two or more, you can form a bridge, and so you see this bridging-induced attraction effect. And the more binding sites you have on the protein, the stronger the effect is.
Indeed, yeah, I remember something like this. I just was thinking about something which is called bond swapping, which is a mechanism that you can find in some classes of polymers, which are called the vitrimers, and that might. I don't know, I was thinking that might be also a possibility for, the swapping of the proteins. But okay, it was just an idea.
Okay, Damian, yeah.
Yes, thanks for the talk. So I understand that your hip hop model depends, I mean, it's controlled by several parameters. No? I can imagine the binding energy, the diffusivities of the different proteins, and so on. So I was wondering how sensitive these structures that you describe, for instance, these coalescence of clusters and so on, how they depend on the different microscopic parameters of your system.
Yeah, so so in terms of the proteins, there are so some parameters are more important than others. So we found that the binding energy tends not to be so important. So long as it's strong enough that you can form a bridge that lasts for long enough time to allow more bridges to form in the same region, then you get this bridging induced attraction. So what is the two parameters which are more important are the switching rate. So these proteins switch back and forward between these binding and non-binding states, and if they switch very quickly, then you get small clusters forming, and if they switch slowly, then the clusters can grow larger, so you can get larger clusters forming. And the other important parameter is then the number of proteins which you have. And so we have done a little bit. So the simulations are quite involved, so we couldn't really do a systematic sweep of parameters, but we've done kind of less than systematic looking at different parameters. And so we varied a few of them, and these are we're using the ones that give the best predictions basically. But those are the two important ones, are the switching rate and the number of proteins that you put in.
Right, thanks.